Hello, in this video we will discuss about the celiac disease. So the celiac disease is the gluten allergy due to the gluten is the protein are present in the wheat and wheat product. And the gluten is basically the gluten after the digestion, the gluten we produce, which that is the allergen for us. So the mouth, esophagus, liver, stomach, and small intestine, large intestine, anus, rectum, appendix, that is all is the digestive system part. So I am recalling again. So in this way the sensitive person will affect gluten, affect the allergic reaction. So how it is possible? The celiac disease is a chronic digestive and immune disorder collectively that damage the same small intestine, the disease triggered by eating food containing gluten, and the gluten contains gliadin after the digestion through a tissue transglutaminase enzyme. So anyhow, here you can see the symptom is the diarrhea, bloating, anemia due to the deficiency of vitamin B12, and growth issue will be. So the growth will be stunt, and not grow too much well, normally the person. So in this way, so let's begin to understand the major activity and how it's work. So here I am drawing the bigger picture of the small intestine, which that's going to lumen and the lamina propria here. You can understand in this way. And lamina propria contain immune cell innate. So anyhow, let's begin to understand. So remember one more thing important. The lamina propria is here and the outer side is the basic lumen. And in this way the commensal bacteria clostridium which that will not infect, and in this way when enter into the lumina propria, so the T regulatory cell induction. Why? Because commensal bacteria saying that T regulatory cell work and please work to stop the immune response. So here is the pathobionts which that will infect the epithelial cell and the epithelial. After the inhibition of the T regulatory cell, the T helper 1 cell activate. So here is the fast ligand and the fast receptor which that will bind, will lead to cascade of phosphorylation, will lead to apoptosis of the epithelial enterocyte. So this is the pathobionts when enter and infect to the epithelial stress, and after the epithelial stress will release the interleukin 15. And anyhow, the interleukin 15 will release, and in this way the interleukin 15 basically recruit the other immune cell, and intraepithelial leukocyte will activate via a HLA-E, MICA, and CD94 NKG2D co-molecule for co-stimulation with cytotoxicity to perforin grain enzyme for the cell death, apoptosis. And here the T regulatory cell will be T regulatory cell induction will be occur. In this way, the T regulatory cell will not induce due to the pathogen will involve, and interleukin 15 will inhibit the T regulatory cell. So the T regulatory cell which that is not work, while the T regulatory cell is responsible for the inhibition of T helper cell. Why? T helper 1 cell. Why? Because the T helper 1 cell release interferon gamma will recruit the dendritic cell, as well as the dendritic cell will release the tumor necrotic factor alpha and interleukin 12 and the innate immune response. And the dendritic cell also recruit through a interleukin 15 for the detection of the antigen from the infected cell also, and the T helper 1 cell also detect through interleukin 21 release from this. So this is the pathogen commensals E. coli, which will also infect, and the mutual relationship containing a epithelial leukocyte will activate again, will lead to damage the epithelium, and damage the epithelial stress and permeability will increase. So the permeability will increase, will lead to pathogen or commensals E. coli. And in this way, the inhibition of the T regulatory cell epithelial dysfunction and permeability increase. So the permeability will increase and the impermeability junction will be separate, and you can understand as well. And this is the inhibition of the T regulatory cell. So the when inhibition of the T regulatory cell, so the T helper 1 cell will be activated, and because the T regulatory cell is the responsible for the inhibition of the T helper 1 cell, and that is the same things you can understand. And in this way, this was the dysfunction and permeability will increase. We are discussing in this manner, and here is I am drawing the line to understand the immune system activation. So that was the immune system activation as much, but in this way, the immune system activation also involved for the damage of the tissue, the damage of the epithelium. But in this case, we are not understanding the innate immune system and the allergic reaction activation. So now we will discuss about the allergic reaction and immune system activation. So here is the dendritic cell, which that is after the detection of the pathogen will release the interferon gamma, tumor necrotic factor alpha, and interleukin 12, which that basically recruit the other cytotoxic T cell to kill that epithelium, the damaged epithelium. So after this, the damaged epitheliums, here is the gluten will enter and can pass. After the passing through entry of the gluten and the TG2, the tissue transglutaminase enzyme used to produce the deamination of gluten peptides, which that's converted into the gliadin. And after this, gliadin will enter into the lamina propria, and in this way, the dendritic cell will detect and engulf this antigen. After this antigen engulfing, go into the lymph node, the lymphatic system. And after go into the lymphatic system, the dendritic cell will become a mature and show the antigen through an antigen representing as an antigen representing cell through MSC class 1 molecule to the CD4 naive CD4 T helper cell. And in this way you can understand the CD4 will activate through gluten specific T cell response to the activate the B cell, and also the activate the T helper 1 cell. T helper 1 cell help to recruit the dendritic cell as well as other immune system and other B cell, more recruitment of more B cell due to the production of interferon gamma and the interleukin. Also the B cell, after the detection of the antigen, we convert by the activation of the CD4 T helper cell to convert the proliferation of B memory cell and plasma cell. So the B memory cell will produce antibody. This antibody is the dimeric, and this dimeric antibody is known as the IgA. This IgA antibody, which that is basically go into also outside of the through permeability, so the outside of the lamina propria, and here is it can be bind to the enterocyte. So the function of this here, so the CD71 receptor are present, which that will be bind with the binding with the gliadin also from the lumen, and after this binding to endocytose, and this endocytosis will be occur, and in this the endosome formation, and this endosome can be neutralized if the normal enterocyte, but if the normal enterocyte will not occur, so it can be neutralized. And here is the endosome formation, and the when acidity will be increased, the pH will decrease due to the influx of the proton. So the junction for impermeability of pathogen, which that will be occur, and the enterocyte here. So when the normal enterocyte, it can be moved into the lamina propria also directly. So when go into the lamina propria, so it will bind with the gluten also. That is the same things you can understand. In this way, the gluten containing a gluten containing antigen, which that will lead to innate immune response activation, meaning that the opsonization will be occur of this gliden antigen, and after the gliden antigen the neutrophil recruitment through the production of cytokines after the opsonization of the gliden, and after this opsonization the neutrophil recruit to endocytose. So this is known as the endocytosis from the neutrophil. After the endocytosis, the digestion lysozymes enzyme, hydrolytic enzyme, and the influx of the proton will lead to form this endosome to digest. After this digestion, the release interleukin and tumor necrotic factor alpha to recruit the monocyte, and this is the monocyte will proliferate after the binding with the interleukin and tumor necrotic factor alpha to proliferate to form a macrophage, and this macrophage which that will engulf this neutrophil is known as erythrocytosis, and this erythrocytosis will cause inflammation, and this inflammation again and again will cause the celiac disease, and this is the function of the celiac disease through a function. Thanks.